आज का टॉपिक मैडम ऑटोमेशन में
हाँ स्टूडेंट सुनते भी हैं
ऐसी है
घर से ही बोलो
मीटिंग में रखो ना ये बात अपनी कि वन साइडेड है। अब वो कर मतलब और क्या अपना सब रूम से अपना रूम से कर सकते हैं, इससे कर सकते हैं, इसमें पता ही नहीं चलता लॉगिन करता अपना
Good morning all. So today's topic is automation in microbiology. So what do you understand by automation? Nowadays everything is being automated. The whole world is going to be digitalized. So we will discuss about automation in microbiology under these headings: background, benefits of automation, hurdles of automation, requirement for automation, equipments and summary. So coming to the background, the role of the clinical microbiology. What is the role of the clinical microbiology? As it assists in the diagnosis of infectious diseases, critical to the patient care, patient outcome, and nowadays infection control. In an era of newly emerging and re-emerging pathogens and antimicrobial resistance, when it is combined with their spread across the continents, microbiology has a key role. So nowadays COVID is also there, so there is so much automation, fully automated machines, and many things are being coming. Historically, there has been a trend towards the automation in clinical chemistry and clinical hematology. This has largely bypassed the clinical microbiology laboratories, because in microbiology the microscopy has not changed much. Manual streaking of the plate is still the same. Incubators are essentially the same. Plate reading has not really changed over the years. What can be the benefits of the automation? Higher total throw output. That means we can increase the number of the samples. More samples can be put, more samples can be reported. Reports can come in a lesser amount of the time. Reduce potential to the human error, as there is subjective error from one person to another person. Limited exposure to the hazardous material, because all of this is infectious material. Any sample which is coming, it is an infectious material. Sometimes you know that it carries this infection, sometimes you don't know. So the person or limited exposure to the lab personnel to this infectious material. Improved record keeping, as everything will be inside the software, so we can improve the record keeping. Reduced labor cost, consistent performance. The machine will give you the consistent performance and less need to find and train the skilled technicians. But what are the hurdles to the automation? As microbiology is too complex to automate a large variety of the samples, including blood, body fluids, urine, catheter tips, tissue, prosthetic devices, and so many variety of the samples we got. Variety of the collection devices, as urine sample has to be collected in urine transport tubes, swabs has to be collected into the viral transport media or another transport medias depending on the which organism you are suspecting, containers for the stool, for tuberculosis another container. So there is a variety of the collection devices. Now variation in the processing of the specimens, as depending on for which test you are applying, there is so much processing of the samples, tissue digestion, decontamination, Even for the same lab, like for the mycology, for the TB lab and for the other labs, due to variety of the samples, the digestion and the decontamination procedures are different, as some samples are sterile or aseptic, but the others are not. So the variety of the digestion, decontamination, smear preparation, some are quantitative culture, some are semi-quantitative culture, so everything is different. So general perception, no machine can replace a human. Critical decision making is required for processing the samples, specifically observation of growth on the agar plates. Machines are programmable but humans are flexible. So no machine can replace a human, because so many times you need to see the colonies, you need to see certain things which you are able to tell, but the machine will tell even a contamination to the possible pathogen organism. The cost of the automation is very high, and the most of the microbiology labs are small, so automation would be underutilized whenever it will be in the place. But there are certain automation systems. What is new? I'm just briefing it. There is a specimen processors, inoculation of tubes and plated media. Specimen processors which can inoculate the tubes and which can plate the media, which can do the subcultures also, plate sticking also, plate labeling also, and barcoding of the samples, slide preparation and staining. So there can be the total laboratory automation systems. Now the automation methods uses one of the following. How do automation occurs? So whenever there is an organism, there is a pathogen, and we know about this pathogen or these organisms characteristics, like it will use carbon dioxide or not, it will use oxygen or not, but enzymes are produced by these organisms. So basically, depending on that, we uses these method, like metabolic end products is the carbon dioxide. So we are going to measure about it. I'll come later on this. Now the enzyme produced by the microorganisms, we can detect that. Oxygen consumption of the microorganisms, heat produced when the organisms grow. Whenever the heat is produced, the organism grows. We can see that. Redox potential is measured, and the fatty acid cell wall components are also measured, because everything is different for every organism, every bacteria, every fungus, every virus. So many things are being a part of how we are going to diagnose in the automation method. Now what is the concept in the technology? All commercial identification systems are based on one of the 5 different technology or a combination. First is the pH based reactions that require 15 to 24 hours of the incubation. Enzyme based reactions that require 2 to 4 hours. So what is that? The pH based reactions, then the enzyme based reactions that require 2 to 4 hours, utilization of the carbon sources, visual detection of the bacterial growth, or detection of the volatile or non-volatile fatty acid via gas chromatography. In pH based reactions, a positive test is indicated by change in the color of one or the more dye. When a carbohydrate is utilized, then a pH becomes acid. When the protein is utilized, there is a release of the nitrogen containing compounds and the pH becomes alkaline. So we are putting basically the reagents which changes their color depending on the reaction. If it becomes acid, like it becomes red or pink. If it becomes alkaline, it becomes blue. So that we can see, or we can counter that this is the growth, that this organism is producing the growth, this sample is producing the growth. Now coming to the enzyme based reactions, color changes in the enzyme based system are due to hydrolysis of a colorless complex by an appropriate enzyme with the resulting release of a chromosome or a fluorescent. So that means one enzyme is being used. So what we do, we put a substrate in that commercial media, we put a substrate. On this substrate, enzyme will act, and from the colorless complex there will be some release of the chromosome or fluorescent, and we can detect that chromosome or fluorescent. So by detection of these methods, we can detect that this sample has a organism or it has the infection or not. Now coming to the utilization of the carbon sources, in this there is a transfer of electrons from an organic product to the dye, that is the tetrazolium violet, which is incorporated within each test well. The transfer causes a colorimetric change in the dye, and these reactions may occur as little as in 4 hours. There can be the other methods like the simple visual detection of the growth of the test organism, which is indicated by increased turbidity in the presence of a substrate. And also the cellular fatty acid metabolism we can visualize. So there is something known as the pre-analytical automation, analytical automation and post-analytical automation. So in the pre-analytical automation, the first semi-automated plate instrument, isoplater, it was developed 23 years ago. Over the year, the more advanced systems have been developed: Innova, Inoquel A, walk-away specimen processor. So automate the plating of the liquid specimens from all kinds of the specimen container. It can be interfaced with the LIS improved efficiency. That means whatever you are plating, it has been recorded into a system in a computer. Read the specimen barcode and label the plated media. So these kind of a isoplater are there. So there can be the uniform staking, and you can see the growth. You can see the individual colonies on it. So this is a unique streaking technology, magnetic rolling bead also, and there is a comb like 16 loops sticking simultaneously. Greater surface area is covered, better separation of the colonies you can see. So next generation incubators are also there. They have the digital camera inside the incubator. So basically what we do in the microbiology, we got the sample first of all, then with the sample we do the processing of the sample depending on the which procedure, which organism we are suspecting, like in the bacteriology, in the mycology, in the TB lab, in the virology. Then after that what we do for the culture, we have to inoculate that sample into the culture media, also depending on the which organism we are suspecting. So when we inoculate into the culture media, after inoculation we put into the incubators. So there is specific timing for the incubator, for the blood culture, for the urine culture, how long, but like 24 hour or more than 24 hour. For TB we detect it, we keep it for the longer time, for mycology also. So these next generation incubators have a digital camera in that. There can be the direct imaging of the culture plates. Incubators can be linked to the plating processing instrument by the conveyor system, and the exact location of the plate in the incubator will be determined by the software. It is the programmed and the timing imaging of the plates. So whenever there is something growth or some image in the camera will capture, it will beep and it will just tell this plate has a growth. So all of these images are stored in the system. Images can be analyzed by the technologist on the computer screen, can be magnified, can be observed under the different lighting conditions. It is better and earlier appreciation of the growth is possible due to the better optics. Images can be compared over the time for the same sample with the other sample or with the previous sample of the patient. If the previous sample of the patient has a similar growth, or the after sample has a similar growth or the different growth, so we can see all these things. And even labs at the off-site location could be assisted by the more experienced ones. Images can be used for the training and it will help in the better quality control. Other components of the automation like blood culture, identification, antimicrobial. This was till the incubation, then the identification of the microorganism. After that, the antimicrobial susceptibility testing. Then coming to the serology, automated ELISAs are also there. Automation of the molecular diagnostic techniques like PCR, polymerase chain reaction, sequencing, DNA microarray chips. Now the blood culture automation, semi-automated systems are approximately 25 years ago. It was BACTEC 225, 301, 460. Latest system are non-invasive, continuously monitoring and hands-off. So there is a non-invasive technology. It is continuous monitoring is there, and it is totally hand off. They can be the BACT alert. It is useful for aerobic, anaerobic and mycobacterial cultures. Like conventional method, about 4 to 7 days, it's a blood culture bottle, conventional blood culture bottle. The blood is inoculated into that. After that we put them into the incubators, and then in the first subculture, then the further subcultures. After subculture, whenever the growth is there, after 7 days we'll declare it as a sterile if there is no growth. Now then after that, if there is a growth, we put the antimicrobial susceptibility, and according to the antimicrobial susceptibility, the drug doses has been decided. So they are standard in the most diagnostic labs. Better detection time, reduce total incubation before calling a bottle as a final negative. So these, whenever the automated blood culture system are there, there's a typical bottles for that, and there is no need of the subculture in between. These automated system will detect the growth. It will give the signal that at this point this blood culture bottle has a growth. So what we do, we take out that blood culture bottle, we do the subculture, and we make the smear and do the staining. With the help of the staining, we are able to tell which organism is there. Is it gram positive, gram negative, cocci, bacilli. Some hint can be given to the clinicians, and antibiotics can be modified according to that. However, with the culture plates, whenever the subculture is being done, so we are more aware. The timing is also reduced in this automation. So better detection times, reduced total incubation time. This was I have telling you. So these are the kind of the images of these blood culture systems. We just put the blood culture bottles inside it. These kind of the blood culture bottles are there. You have to just put the blood into it and incubate it into these systems, and automatically they will detect there is a growth or there is a growth or there is not a growth. So Bactec vial. In earlier system, what is the principle of this system? In Bactec vial, in earlier system, the bacteria if present, it utilize the radio labeled 14. What is the carbon number in the natural? It is the 12. So if 14 carbon labeled substrate, and it releases radio labeled carbon dioxide. So this instrument measures so quantitatively the radioactivity on a scaling range from 0 to 999 as a growth index. So the daily increase in the growth index is directly proportional to the rate and amount of the growth in the media. So newer instruments measure the carbon dioxide content by the infrared analyzer. This is the BACT alert 9240. It uses a calorimetric photosensor. So there is a photosensor which is calorimetric, and the reflected light to monitor the presence of CO2 dissolved in the culture media. So whenever in the sample a organism is present, it will use the carbon source and it will release carbon dioxide. So how we are going to detect these carbon dioxide? This is the basics of this automation in these cases. The CO2 sensor inside the bottle is bonded to the bottom of the bottle and separated from the both by a membrane that is freely permeable to the carbon dioxide. So whenever carbon dioxide is produced, the color in the sensor changes from dark green to the yellow. Now coming to the mycobacterium growth indicator tube, it is known as the Midget 960. It is used for the tuberculosis, as its name suggests, mycobacterium. It consists of 16 to 100 mm round bottom glass tube, and it contains broth, that is a 7H11 broth base, to which is added 0.5 ml of OADC, oleic acid, bovine albumin, dextrose and catalase, and 0.1 ml of the antibiotic mixture to inhibit the contaminants. Now what does these four things will do? Oleic acid is a metabolic stimulant of the mycobacteria. Albumin binds toxic free acids. Dextrose is an energy source. Catalase to destroy the peroxides. A fluorescent compound is embedded to the silicone at bottom of the tube. Oxygen in uninoculated medium serves to quench the fluorescent light. So whenever there is an uninoculated media, there is an oxygen which will quench the fluorescence light. So this is the midget system. These are the midget system, and these are the tubes. These are the uninoculated tubes. Whenever there is uninoculation, the oxygen is present in the media and it is quenching the fluorescence. So whenever bacteria is present, the mycobacteria is present in the sample, it grows. So when it grows, what it will utilize? It will utilize oxygen. So when it utilizes the oxygen, what happens? The quenching of that fluorescent compound will no longer be present there. So fluorescence, you can see or you can measure the fluorescence in this fully automated compound. As actively growing bacteria consume the dissolved oxygen and the fluorescence is unmasked and it is detected by UV light. So cap is replaced and tube is inverted to mix the component. These are basically the mechanism how we are going to add this OADC. And tubes are read every other day starting on the second day with a word slam. Positive tube is when a bright orange color is seen at the bottom of the tube with an orange reflection at the meniscus. So they are concerned with all these automated systems also. False positive due to high TLC count, continuous power supply is needed. Cost is also an important factor, like in the peripheries, the continuous power supply, the trained skilled technicians to handle all these things, and the supply of the blood culture bottles is an important. So now coming after this, so first of all it was the pre-analytical, then analytical, how the culture are being done in the machines. The second point, how the identification is being done in the machines, in the fully automated systems. We do have the conventional ID and antimicrobial susceptibility testing. So from the culture, whenever these automated system has given a beep, we had put these culture plates or we had done the subculture and we can see the growth. Now from the growth, what we do, the conventional biochemical reaction will put to identify we'll do the gram staining also, microscopy, and then the biochemical reactions to see which organism is there, and then antimicrobial susceptibility testing is being done. Now coming to the automated ID and sensitivity system. Automated ID testing is available since 1977. Biochemical results are read by calorimetric or fluorimetric means. Cards or panels of the antibiotic in different dilution to ascertain the MICs, that is a minimum inhibitory concentration. It is available in many labs. Advantages of automated ID and AST. There is a reduced errors, improved reproducibility, as the same subculture should give the similar antimicrobial susceptibility testing results. Improved ID is linked to the AST, improved speed of the result, increased number of the agents. Like when we had we done the biochemical reaction, it will take another day to identify the organism, now the another day to identify the subculture antimicrobial susceptibility testing. So with these system we can improve both of these things. We do have the available system Vitec, Biomeru, Microscan walkaway system and Phoenix BP system. In Vitec 2, the ID identification currently is biochemical. 30 wells with the biochemical substances dehydrated forms. And Vitec identification covers over 300 species. AST is based on the broad susceptibility with specific cards. These cards does have the 30 or the 45 wells containing dehydrated antibiotics, and the specific tests are there for detecting resistance mechanism systematically. So time to result for ID is 2 to 10 hours for bacterial isolates and approximately 18 hours for yeast. Time to result for AST is 2 to 18 hours. Like coming to the Microscan walkaway system, it is a 96 well plate system. What we do, AST is based on the conventional micropore dilation, that is minimum inhibitory concentration, and ID is currently biochemical. Growth and reactions are continuously monitored in this. So what is the difference between these three systems, AIITech 2, Microscan and Phoenix? What will it will detect? IB, G+, anaerobic, yeast. So gram positive, gram negative, anaerobic and yeast. Gram positive, gram negative, anaerobic and yeast. And the Phoenix only gram positive and gram negative. Identification. Now coming to the antimicrobial susceptibility testing, gram positive, gram negative and yeast. Gram positive and gram negative only in Microscan. And Phoenix is the gram positive and gram negative. Reading is the continuous and automatic. Microscan it is a continuous, automated, also manual. It is continuous and automated. So yes, these are all three biochemical ID. Plans for integration with known biochemical system, yes. Epidemiological software, yes. Cost for comparable model for 60. Test it cost around 25 lakhs, 20 lakhs and 40 lakhs. What is the problems with antimicrobial susceptibility system and identification system? We need pure culture for that. If any mixture will be there, it will give very erroneous results. Simple tests are not covered like oxidase, catalase, motility and the pigment, which is also have the contribution in identification of the organism. Variability in identification of the rarer organisms. So very rare organisms are also reported on these platforms, and we don't know we are truly reporting these pathogenic. Is it pathogenic or is it something else? It depends on the database available. It means this system, this software, this computer has all the data of these organisms, of these their sensitivities, and all of these data is there, and according to this data the software detects this identification and antimicrobial susceptibility. Anything outside this data will not be recorded and it will not resulted, it will not reported. So no identification, no antimicrobial susceptibility. Also the inoculum size can affect the MIC interpretations. The inoculum size what we have used before inoculating into these plates, 96 well plates, so it will also result in these things. Now coming to the MALDI, this is the matrix assisted laser desorption and ionization. The method where laser is used to generate the ions of high molecular weight samples such as proteins and polymers. So these are the newer systems, matrix assisted laser desorption and ionization. Its name is itself telling you what it is. The method where a laser is used to generate ions of high molecular weight samples such as proteins and the polymers. So in a sample we do have the proteins, we do have the polymers. So a laser is used to separate these high molecular weight samples which contains proteins, and these ions are generated and we can identify according to the database which ions are these and from which organism it does belong to. Absorption of light by the matrix, then transfer of this energy to the analyte, ionize into the gas phase. High electric field is then used to accelerate these resulting ions into the flight tube in the mass spectrophotometer. So this is the sample culture is there. We have the growth. From this growth, this tube we had put the inoculum and there is a matrix. This matrix has been inoculated, like this is also the plate A, B, C, D, and this one is a. So we had put at the 5th number we had put the sample matrix into that. In the system the light will be there, and the light will be there. It will create the energy and it will change into the ions. These ions, the laser is there. This is the electrostatic field. So ions will be released. These are the proteins and the matrix. These proteins and matrix, this is the time of the flight according to the mass ratio, energy ratio. These ions, how these are dissolving into this, how these are moving or revolving to the time of the flight. So there is a mass to charge ratio which is being generated from this tube, and the graph is being plotted. So this graph of mass to charge in this spectrometer will be detected and will be matched to the database. Every system, every automated system has a database of the organism of identification or antimicrobial susceptibility, and according to that it is matched and the identification has been given, like this is the organism. So what is the use of the system is like, we just have the sample, next day we had done the identification and antimicrobial susceptibility for this one also. As we have not wasted our time, within 3 to 4 hours of the growth we can give the results, but again its limitation is a similar one, as whatever is in the data we can give the result, we can report. Protein, so it depends on the protein profile based identification. It's mainly used for bacteria, also for yeast, filamentous fungi and mycobacteria. Rapid identification in minutes. More than 90% of all isolates are identified at the species level. 98% are identified at the genus level. So, and less than 1% are incorrectly identified. So we can rely something on that. So this is Malditov, also being introduced in the virology essay. Suppliers are the Biomeru and Bruker. Now what are the problems with the Malditov? Still not directly applicable to the specimen. We need to have the culture, pure culture. In mixed culture it gives unacceptable results, and for accurate identification we need a good CFU, that is 1.5 into 10 raised to the power 5. It is not useful for viruses. Identifies only organisms in the database. Antimicrobial susceptibility and the strain typing may be possible in future. Automated immunological testing is also there. It depends on the VIDAS system. These are the various systems. One is a VIDAS, Biomeru and Evolus BioRed. VIDAS is the principle of the enzyme-linked fluorescent assay. It detects antigen and antibodies for various infections, diseases including viral hepatitis and torch. There is a benchtop automated pipetter, incubator, washer, reader for the enzyme immunoassay microplate assays. Advantage is the rapid result. Automation has an advantage of the rapid result, no carryover contamination. Now coming to the molecular methods, that is the automated PCR system. Automated or sample to result open and the closed platforms are available for this. Abbott is there, Cobas system of the Roche is there. So this is the Cobas Amplicur system, was the first fully automated system 1995 for PCR routine diagnostic. Now this Cobas Amplicur combines thermal cycler, automated pipetter, incubator, washer and reader into one system. It can identify HIV-1, HCV, hepatitis B, cytomegalovirus, chlamydia, Mycobacterium tuberculosis. Now coming to the Gene Expert. Gene Expert is also a fully automated system, and is a semi-quantitative nested real-time PCR is there. Not only for tuberculosis but also for MRSA, Clostridium difficile, HIV, HCV, HPV. Nowadays they are increasing their things. Expert MTB RIF is an automated cartridge based nucleic acid amplification assay for simultaneous detection of the TB and the rifampicin resistance directly from the sputum in 2 hours. Sensitivity and specificity is more than 90%. So Expert MTB is from CAFED. It is an automated cartridge based. This is the cartridge, and we need only, we basically need only sample. We just put the sample into it and just inoculate this cartridge into the system, and after 2 hours we can get the result. Mycobacterium tuberculosis is present or not present, or it is rifampicin resistance or it is not resistant. And the second point is that basically they are also increasing their domain about the first line drugs, second line drugs also. And there is something known as the TrueNat is also there, which is nowadays is more likely, or you have heard about this COVID diagnosis, TrueNet is a role. So TrueNet is also a molecular method. It is also on the similar principle of the PCR, that is a real-time PCR is also there. It is a smaller machine. Has only it has a very smaller machine. It doesn't need the AC and the other electricity ones. You need to just charge it and you can carry it to the peripheries also. So this can be a good option along with the gene expert. Issues with the molecular methods: it cannot differentiate from live bacteria or a dead bacteria, because they are just taking out the DNA or RNA of the organisms. They don't know it is a live or it is dead one. They'll report both. Negative results also do not rule out the infections, because sometimes there's a less bacteria or less organism from which DNA we cannot take out, or some mutations are also there. So our conventional methods are, at that time it plays a good role. Now coming to the sequencers, nowadays we do have the next generation sequencers. Into the whole genome sequencing is being done from the organism. We have that culture plate is very important for us in microbiology. What we do, we just put the sample into these kind of the systems. These are the Illumina for the next generation sequencer. It will sequence whole the bacteria, it will sequence whole the DNA or RNA, and then the sequences, there is a database. Into that we can match these database and we can identify the mutations from the bacteria. These mutations are important for identification of the antimicrobial susceptibility, antimicrobial resistance, detection of the new mutations, and the which mutations are important, which is leading to this resistance. And these resistance are basically very bad scenario in the future, in the coming world. So all of these things are very important. Regarding from the point, like conventional is also important, microscopy is also important, culture is also important. Automation has its own role in certain way to decreasing the turnaround time, to give the effective reporting, to report the clinicians. So now coming to the summary, the automation can result in efficiency and the personal cost saving. It allows for the standardization, like the one sample should give the similar result either by me, either by someone else who will put the sample. Total laboratory automation occurs when the several automation systems come together. However, it is very less feasibility in India. We do have very smaller labs also in the peripheries also. But like from the as we can say, from the tuberculosis program, the gene expert is available at many places. Midget is also available at many places. So many RNTCP labs are there. So according to this, we can say that at least the important machine should be there, or important fully automated machine should be there even at the periphery level, to give the good reporting, and so that the reporting can be given in a time and the patient can get a good or better or proper treatment, good antibiotics depending on their sensitivity. Necessity and the extent of the automation is required independent in the lab. Further studies are needed to understand the clinical impact of the automation on patient's outcome and the clinical lab workflow. Thank you, but automation will never replace us. Thank you so much. इनका अटेंडेंस तो आया हुआ है। आपने पेन ड्राइव में चलाया था ना डायरेक्ट?